We'll start. Good afternoon, everyone. It's my pleasure to welcome you all to today's webinar titled Orbiting Innovation, How Space Research is Transforming Global Health. My name is Sridhar Narayanan. I'm the Senior Technical Advisor for Innovation for the Indian Pharmaceutical Alliance. I'm honored to be moderating this session today. As we stand at the intersection of biotechnology and space sciences, we are beginning to explore frontiers that have remained largely unexplored to date. How the unique environment of space can be leveraged to accelerate pharmaceutical innovation is a question that we will try to answer today. From protein crystallization in microgravity to understanding disease mechanisms in extreme conditions, space-based research is already beginning to shape the future of healthcare. Globally, pharmaceutical research in space is making great strides, and India, with its world-class pharmaceutical industry and an increasing space presence, is poised to make a huge impact in the world of pharmaceutical research in space. Our Honorable Prime Minister, in his Mann Ki Baat address a few weeks ago, also brought out the importance of space research to the economy in India and definitely to the pharmaceutical industry. Today's session brings together researchers, industry leaders, and policymakers who are experts in this area to discuss the scientific potential of space research to the pharma industry. Thank you all for joining this webinar. Just a couple of housekeeping notes before we get started with our first speaker. Feel free to put your questions in the chat box. We will have a question and answer session after all the panelists have spoken. Thank you very much. And now it is my pleasure to introduce you to our first speaker, Dr. Adeel Nasir. Dr. Adeel Nasir earned his PhD in the field of gravitational biology in Germany, and his PhD thesis was focused on deciphering the molecular basis of light and gravity sensing mechanisms in the unicellular organism Euglena gracilis. He had the chance to send his experiment in low Earth orbit on board a non-manned spacecraft in a Chinese-German space mission. He is the proponent of the initial idea of a biological life support system for space, which can orbit around the Earth for over a year on a satellite mission. Based on his diverse training, he is convinced that space environment can help humanity to develop useful technologies. In his own words, space technologies will not only help humans to colonize space, but also tackle severe challenges on Earth. It is my pleasure to welcome Adeel once again. Nikhil, can we have the first slide, please?
Thank you very much for the kind introduction, Dr. Sridhar. Okay, so let's get started. Hello, everyone. I'm going to talk about how spacefaring is helping us to improve human health on Earth. So my name is Adeel Nasir. Dr. Sridhar has introduced me very well. I don't think I can do better introduction, what he has done already. So moving further, can we go to the next slide, Nikhil? So you may call me a rocket biologist because I have been to different kind of projects that involved gravitational and light sensing mechanisms for unicellular organisms. This was my first interaction as my PhD thesis. And I had a chance to work on a German and a Chinese space station combined project, which involved the coupling maneuver of Chinese space station in the beginning. And I was able to send my experiments there. And I proposed this idea for this Eucropus. This is a Eucropus is a biological life support system that has orbited the Earth with NASA on the same satellite. This was my second contribution to the space industry. And here, this is me. If you can play the video, please, Nikhil. So yeah, I'm performing experiment in the parabolic flight, studying the gravity sensing mechanism. In addition to that, I did a postdoc in the field of drug development where I was trying to understand how drug repurposing, the FDA molecules already approved, already existing, can be used to fix Parkinson's disease, which is a second lethal disease in the field of neurobiological diseases. And I also worked on the antimicrobial resistance bacterial strains and trying to fix, or I'm trying to eradicate those bacteria that exist in the cystic fibrosis patient's lungs. So basically, this is the brief introduction of my space career. Moving further, can you go on the next slide, please? So currently, I'm involved in different projects across the continents. These are my intercontinental engagements. I'm working with a company called SpaceBorn, and they have a dream to produce human baby in space. And there I'm working for the development of mini lab for assisted reproductive technology. In the word of my CEO, he says that life need a plan B because war and the environmental changes in Earth may not keep the Earth intact, and we need to go towards stars and find our way to seed our life. And my other intercontinental project is that I'm engaged with ResearchSat, a company that believes that every microgravity experiment makes a world with a better, healthier place on Earth. And I have a show, this is called Rocket Biologist, and I there talk to experts in the field of space biology, and it's my mission to create a new breed of space biologists across the world. So this is, I'm on the mission. So going further, here I have a story to tell you. This is the story of human civilization, our mankind story. The year is 1600. A country made a company called British India Company, and they sent their vessel called Red Dragon to find and do trade to a rich country. At that time, it was not sure what is India. It was being confused by Indonesia and India. So they came primarily, the ship reached to Indonesia. There they did trading, and eventually they finally reached to the shores of India. This was the first connection of a foreign people coming to India. Along that process, I'm not going to talk about that, what are the implications of that process. A British doctor was studying medicinal plants in India. So seafaring eventually helped people to see medicinal plant of across the continent. So basically, in other words, medicine is being progressed when humans start exploration. Moving further to the next slide, please. So the same spirit that humanity is keeping, and now the same species are trying to go beyond seas. They're trying to jump into space and outer space. Unlike the place where they were, the beautiful place of India, they will not have that beautiful environment to survive. This is an hostile and extreme environment, but the motivation to go there is the motivation to get resources. So that's put the end to that discussion. This is not fiction. It is really a fact that people are going to space to exploit the resources of those places. And along with that process, when we are going up there, we need to protect humanity. We need to protect human health. So the next slide, please. In that process of going to space, there are different technological advancements that are taking place. But one thing that remains irreplaceable, that is biology, the knowledge of biology, the flexibility of life, and how it can be modified and how it can beautifully adapt to different extreme conditions. This is something getting attention these days. For instance, that in the last few decades, the use of biology in different forms in the space exploration has increased. And there's a very wonderful book written by Christopher Mason. He's suggesting to use life, to engineer life, to survive intergalactically in the space for 500 years. So this is like a vision he's trying to present, and the biology and the field of medicine remains at the forefront of that exploration. So similarly, if you want to have a habitat up there, you need to find a sustainable solution, and the life gives that very easily to you, because life has an ability to adapt in different sorts of conditions. So the tool of biology, knowledge of biology, will pave our way towards stars. Please, to the next slide. So what's the learning from space? So far, we are standing here. We have done, this is the collective human, irrespective of theatrics of space, this is the collective human knowledge that has been displayed in the outer orbit. And these are called those space stations. And from those space stations, humanity has learned a lot. But this has to be understood that we are standing under, we are just not far from Earth, and we are protected by the magnetic field of Earth. Once we go out to explore Mars and Moon, we are going to be exposed to another level of radiation, because our magnetic field of the Earth is protecting us. So moving further, we don't have a long-term knowledge, long-term data, how human life would react to that, or how life would react to that. So this is an open question for scientists. And the learnings, the teachings that we have from the space, going to the space stations, how biology react to that. In the next slide, I'm going to tell you about that briefly. So this is how it looks like, that the Earth-based life on different kind of formations, that microbes, plants, or human, what happened to them once they are up there. So for human, this is a very drastic impact that the human body have. A lot of biological systems, a lot of major systems are disturbed, and they have huge problem to survive, to stay there for long term. In case of plants go through the same process, but they are better, adapting better to space conditions, depending on different species to space. However, the microbes stand out all those challenges, and they stand like a champion. The adaptability is higher, much higher, not to exaggerate, but they can adapt much better. And there are a lot of studies that shows that the resistance bacteria on International Space Station has been found, which means that they were able to handle the space conditions better than anything else. And the impact at the cellular level are huge. What happened that in the microgravity conditions, the heat distribution in the cell, gas distribution in the cell, nutrient availability, it changed. And ionization is affecting the DNA. So the damages are greater. So now the question is, going to the further slide, why the hell on earth we are going up there? So let's say when you're doing something wrong, do it right. So to countermeasure that, what is happening there? The space environment is filled with space radiation, altered gravity, isolation, confinement, closed environment, and distance from Earth. There are different strategies that humans have come up with to navigate in this extreme environment. For example, that one of the recent technologies that they are trying to bring holographic images or holograph transportation of holograph medical doctor to International Space Station, so the astronauts can feel more connected. And this is one thing. And then the list of possibilities is huge. Miniaturization of equipment, 3D printing for not only for the material, for the human, for the life, and then other kind of equipments that one can think of, the use of CRISPR-Cas or the technology in the molecular biology to improve human life. So this is just the gist of it, that what can be done. So on the next slide, please. So if we are trying to get into the field of space medicine, space biology, There are a few areas that I can come up with that, for example, that we have to start being curious. We have to be creative and take it to the commercialization. So in this process, if you look at the drug development process, a lot of diseases, a lot of molecules has a crystalline structure. I think Antrix has a lot to talk about that on this subject. They in the microgravity environment, the formation of crystals are much better. So we can understand how this particular molecule is going to behave, how it's going to interact with cells, and that can help us to eradicate or to navigate through how to find different kind of drugs for different sort of diseases. So this is one of the area that has always been there, and now it's getting a lot of attention. And the second area that is drug repurposing. Space provides you with this extreme environment that can be used for drug testing, drug analysis. And there's a recent paper, which is like still at the very beginning, that a medicine that is used for curing diabetes, it's now turns out it's a radioprotective, and this study was done in the NASA radiology labs. Another most interesting subject is biofabrication, and that is the production of organs in space using this technology that is already Earth-based stem cell research. And this sky is the limit. Organ is one thing, and people are trying to make food, like, for example, the meat in space, which may not be a bad idea to survive. So basically, this 3D printing or stem cell research is being exploited on different fronts, including the space medicine. And then the microbiome and antimicrobial research. A lot of studies have shown that when microbes are exposed to space conditions, they behave very differently. The replication time is shorter. They're resistant to different kind of space environment. And there is a company that is working, it's called Astrobiome, on two sorts of studies that they're trying to how do I say, strengthen the microbiome by bringing them to space and bringing back on Earth. And they are trying to use it for agriculture as well as for the medicine. So this is the area that we can think of. Moving further, please. So what I am talking about, is it all science fiction? No, it's not. It's happening, and there are commercial players which are doing that. So one of the shining examples of this is the drug Ketrida, and I think again I would let the other speakers to talk about it. This molecule was studied. This is a monoclonal antibody. The structure was studied on International Space Station, and once it was studied, the structure that was formed there, the crystalline structure, they were able to improve the administration of that drug in patients differently, and that was a big breakthrough. Based on that, there is a different kind of companies which are in this space medicine research are growing, and there are some big giants already, giant pharmaceuticals, and there are new companies coming up. So the industry, the space biology or space medicine industry is booming. And there is one of my favorite companies also in the list, so I thought to highlight it, and that is ResearchSet. The next one, please. So now the question is who and where we can, how and where we can perform those experiments. There are different platforms for the microgravity research. There are Earth-based, which are called like ground control systems, which can simulate microgravity for short term. And there are some sounding rocket flights, which are they goes in the low Earth orbit, and they stay there for a few minutes and come back. And there is a drop tower in Germany that can be used for studying microgravity. And then there's a long-term real microgravity platforms where these researches can be performed. And I think all those platforms, in the past, they were very expensive. I mean, it's a relative statement to say. When I started working in the field, at least I saw that, it was really not easy to access. But now they're getting very accessible. They're getting cheaper. So this is the advent of low Earth economy, the low Earth orbit economy. So people are bringing their experiments up there. So the next slide, please. So in the end of the day, that human exploration has taken further. So if you have the next idea about that can be used in the space, so I think the Pegasus of research research set is there to bring your idea to the next stage. And for that, I think Jibin is the person of interest you have to talk to if you have an idea that you think it can go up into space. And he has a lot to offer: the active experimentation, passive experimentation, cell bank, and in space manufacturing. So on the last slide, please. So I would conclude with that space can be sometimes dull. So let's keep it colorful by bringing people all across the world. And we really don't have to reinvent the wheel. We just have to spin it differently. Thank you very much.
Thank you, Dr. Adeel, for a wonderful talk. We will take questions at the end of the session. I'd like to introduce the next speaker, whose name, in fact, is a translation of space, Antriksh Paricha. Antriksh is the CEO for a company called Serendipity Space, which is a startup based out of India. He has a degree in a master's degree in applied and engineering physics from Germany. He has built levitation systems at Europe's first passenger scale Hyperloop pod at the German Hyperloop program, and has worked on several projects related to space and pharmaceutical innovation. Serendipity Space as a company wants to produce pharmaceuticals in the low Earth orbit and bring them back to Earth to accelerate the drug development in the Earth situation. Antriksh, over to you. And Antriksh will be talking about the chemistry aspects of space research.
Yes, thank you so much, Sridhar sir. Hi everyone. This is a great platform to be speaking at the intersection of multiple sciences, so to say. So hence I've titled my talk "Beyond Earth and Beyond Limits" because that's really where we are going to. My name is Antariksh. I'm the co-founder and CEO of Serendipity Space. And Nikhil, can we move to the next? Yeah, and our mission is to basically accelerate drug development by producing some of these pharmaceuticals in space and then bringing them back to Earth. So for that, we are actually building small reusable satellites which can process these pharmaceuticals in space, spend a couple of months over there, and then also return on their own. So we are going for a fully unmanned approach to doing this because sending humans, as important as it is going to ratchet up the cost of any drug development. Next. So right now, this is the staggering statistic that we are working towards. The status quo has been that currently the drug development of most pharmaceutical companies around the world, or I mean, all frankly, pharmaceutical companies around the world, takes a long time and costs a lot of money right now. Next. Yeah, currently the time taken is anywhere between 10 to 15 years to develop a new drug, and it costs anywhere between $2 to $3 billion per drug. So to bring it from its birth to its maturity, and even then the failure rate can be as high as 90%. Anywhere you never know where the drug might fail, and the reason for that has to be that we have to understand what is the scientific problem that actually leads to such a high cost and high amount of time that is taken. So here are the few list of problems that usually happen in some of the problems that happen in a pharmaceutical development cycle. The one is unsuccessful crystallization. So this can be of the protein that you're targeting as well as the therapeutic that you're making as well. Now here actually a lot of the failures encountered here are work with a patch, like patch-like framework where you have to do multiple experiments, multiple research, and long-term R&D to actually figure out what the structure of your drug compound is or what the structure of the protein is, so that you can actually design a drug based on that. And apart from crystallization, there are things such as uniform particle size. You're not able to get particle sizes which are uniform in size. There's also solubility issues sometimes, even when you get to the formulation stage. And all of these actually lead to repeated trial and error. And that's where actually a lot of the money and a lot of the time goes. And the thing is, Nikhil, if we can move to the next slide, pharmaceutical companies have had a secret, at least in the West, where they have actually used pharmacy, the space as a platform to bypass some of these problems. And this is the industry secret that there is, that due to absence of sedimentation, absence of convection currents, and absence of buoyancy, the absence of basically gravity leads to the absence of these fundamental forces as well. And as a result of which, you actually end up with much better drug products formed. Now, it's not only true for pharmaceuticals, by the way. This goes for any other compound, any other material that you produce as well. You often are able to get much higher purity when you produce it in zero gravity. Next, Nikhil. Yeah, so now I'm going to talk about what can be done, frankly, pragmatically in space for pharmaceutical companies where we are at the commercial stages, what can actually give you commercial returns as well for a company. Now, if you look at microgravity pharmaceutical production, the history of that in the ISS or in the previous Star Labs or the Mir space stations by the Russians, you can see that there are fundamentally, you can club them into two separate boxes. One is micro scale and one is macro scale. So where you produce a small number of, small quantity of pharmaceuticals, and one where you produce a large quantity of pharmaceuticals. Now, when I say pharmaceuticals, it could mean the proteins that are targeted as well as the therapeutics that are being made. Now, in case of micro scale, how does it exactly translate into value based, something that of value for terrestrial markets, is that when we make some of these proteins, which are very novel proteins, so to say, which are not usually being crystallized on Earth, you can actually open up new drug discovery pathways. So which directly means that you can actually develop new drugs based on it. So when you do something like, and this does not need something like a lot of kilogram quantity, this can be done in a few grams, and then you can bring back the crystals, you can study them, and based on that, you can do something known as structure-based drug design, something that Sridhar sir knows much more about than frankly all of us. Because this is the core concept of AI-based drug discovery as well, where you actually have a rational approach to actually designing drugs. And the second aspect is the crystal form selection. So let's say you have developed a pharmaceutical. Now you want to see which crystalline structure would be suitable for the dosage as well as for the particular efficacy that you need in as a formulation. And that is where you can select which crystalline form. And one of the most famous cases is ritonavir, which was actually called back from the market in 1990s when it crystallized in a different form in patients' bodies. And now it is finding new avenues in microgravity to actually introduce a different kind of a crystal form which is stable in the human body. And in case of macro scale productions, these can be exactly as Dr. Adeel also rightly mentioned in his talk. One of the famous stories was of Keytruda, where you can do actually macro scale production of these pharmaceuticals, which means that you can produce the APIs of the drug compounds and both small molecules and biologics. And clearly, at least, we have seen development like some breakthroughs in biologics for sure. So this can lead to high dosage concentration, something like subcutaneous injections, which you can maybe sit at home and take it instead of going to the hospital, as well as enhanced formulations. Next, maybe. So now what I'll do is actually I'll tell you where we are in the commercial sense of things, like what commercial successes have happened, and how do you exactly ride that progress and actually produce something which is business-like and meaningful. So this is one of the companies called Karna Biosciences, which is in Japan. And they actually were not able to get crystal structure, which is of a very high resolution on Earth of the MAP 2K7 protein. This is a very important protein for any cancer drug discovery program. And by going to space, they were actually able to make a very high make a pure crystal, which resulted in a high resolution structure of that protein. Now, having that high resolution structure actually leads to much quicker drug discovery. rather than doing, multiple trial and error. So your search space from compounds actually reduces when you have a very good high resolution structure. So, and these are not my words, but some companies have told us from our customer interviews as well that they can save anywhere between 4 to 5 years if they had actually really good structural data, which is below a certain angstrom size, angstrom resolution. So this was one of the successes in case of the micro scale production, which I said, which is like a few grams of crystals that you can produce. Next, Nicholas. This is one more, a company called Taiho Pharmaceuticals, which actually produced a drug out of crystallizing in space for the HPGDS enzyme. This is a drug for Duchenne's muscular dystrophy. This is currently in its phase 3 clinical trials. So you can see that this is directly translated into commercial successes, even if you make a few grams of it in space. So that's the value that this holds. Can we move to the next? Next, please. Yeah. So now we talked about if we produce few grams of crystals, we can actually accelerate drug development. But when we do something like a kilogram scale production, we can actually go up to producing APIs in space. And the right examples are Merck's Keytruda, where they were able to actually obtain a subcutaneous administration of a drug where they could control the particle sizes, the viscosity, and the unique problem to solve is that when you're doing a protein therapeutic, when you increase its dosage, its viscosity also increases. That's where the problem lies, and they use space as an environment to bypass that problem, where you can increase the dosage, but the viscosity doesn't increase, and it can remain stable in during injection as well. And this was based off of a previous experiment which was done in the late 1990s by Schering Plough for interferon alpha, which actually they tested in cynomolgus monkeys as well, and this has remained as a stable formulation over 2 years after coming back from space. So there is definitely macro scale production successes or indications that this can happen. And this is where micro and macro can have like a synergistic relationship where we can do multiple customers based on the needs. Can we move to the next? And so we actually designed our first pharmaceutical labs, so to say, the autonomous labs, which can do protein crystallization or small molecule crystallization. And you can see that in the image as well. I can also hold it up if you can see my screen. This was like a smaller version of it. I don't know if you guys can see it. This was like a smaller version of it where we actually crystallized protein therapeutics and small molecules as well. Next, please. This is primarily for protein crystallization. We did one for formulations as well, where you can actually, first of all, do proof of concept studies for a few ml, few milliliters of the formulation. And once you get something which is of value, we can scale it up to kilogram scales as well. Can we move to the next? Now, what are we exactly doing? What are we exactly building? And how do we see the future of something like space-based pharmaceutical production become a reality? Is that we are making satellites which are currently about the size of a travel suitcase that you usually carry around. This can be launched to a low Earth orbit. It can spend a couple of months in orbit, make these pharmaceuticals on a small scale, and then also bring them back on their own. And then we have a soft landing basically somewhere close to the ocean, and then we retrieve the drugs and supply it to pharmaceutical companies. That's basically what our mission plan is. Next. And this here is the team. Jivitesh is the CTO and co-founder. He's a master's in robotics and AI from Rochester. He handles our automation and manufacturing. Dr. Monica Eckel, she previously worked at as NASA and German Aerospace Center. She's handling our space system side of things. And lastly, me, I have a degree in applied physics. I worked at the Hyperloop program and a few US Department of Defense projects. I handle the business development and the tech roadmap. Can we move to the next? So on the pharmaceutical side, we actually have Dr. Subhabrata Sen, who previously served as the senior and scientific manager at Syngene and Pfizer India, also at GVK Bio, and Dr. Parthapratim Munshi, who worked at the Oak Ridge for protein crystallization on the ISS. Basically, we make our hardware, we test it in the labs, and we produce pharmaceuticals. That's where we are right now. Can you next? Yeah, that's about it. Would love to know your questions, and thank you so much.
Thank you, and Antriksh, for that excellent summary of what kind of chemistry can be done in space. Participants, you can put your questions in the chat box, and we will have a Q&A session at the end. Now it's time to move from chemistry to a more biology focus and look at how space research is affecting biological systems. I'd like to invite Dr. Suresh Poosala, who's the founder of OncoSeq Bio and also an advisor to our side space research. Suresh is a very well-known figure in the pharmaceutical industry, not only in India, but also in the United States. He has established several preclinical facilities for Syngene BBRC Biocon, both in Bangalore and also for BMS in Redwood City, California. He's a veterinary science graduate from Tirupati and has a master's in microbiology and immunology and a PhD from the Iowa State University. He's also the first council member on a triple ALAC international accreditation from India and serves on the executive council of the Federation for Asian Biotech Association. Suresh is an well-acclaimed scientist in the field of 3D printing and also in the field of 3D organoids. Suresh, over to you to talk about biology in space. He'll be immediately followed by Jibin, who's the co-founder and the chief technology officer for RSAT Research, which is an aerospace company based out of Australia and also has a presence in Hyderabad, India. He's an aerospace engineer by training and has a keen interest in microbiological and pharmaceutical research in space. He leads the technology development for advanced scientific payloads for industrial and university clients and oversees end-to-end mission management process with rocket launch partners, regulatory agencies, and with the relevant authorities to undertake microgravity research. He has so far led 2 suborbital rocket launch flights from Sweden and is also the co-founder for RSAT Space Technology based out of Hyderabad. He wants to bring these deep technology capabilities to India to improve the way space research is utilized in India and to improve the kind of consumption of such technologies in the low and middle income countries. Over to you, Suresh and Jibin.
Thank you, Sridhar. Wonderful introduction. Graceful, thank you. I think I will spend little time on what is being done on the ground. Thanks to Adeel and Antariksh, they have covered part of the program that I wanted to present to you on, especially on the crystallization and Keytruda. So that's a good story, a successful story, and great learning opportunity for me as well. So in biology, on the ground, what I do here is build 3D organoids. I collect the tissues. Why 3D? Because that's the real-time replication of organs and cells within the body. Nothing is on a planar model. So they actually form 3D structures. So we create round balls of primary cells or tumors from patients or cell lines and other primary cells. So that's what's happening here. And then from the space perspective, what we plan is we want to see how a cell behaves, how these interactions of multiple cells happen on the ground in comparison to space. And there are a lot of advantages of doing this in the space. And what are those? So some of those things we are going to evaluate. Next slide, please. Next slide. So we are able to fight cancer, and Keytruda is one of the primary drugs that has achieved it primarily from BMS as an Opdivo, later on from Merck as Keytruda. And so it's a very successful drug. So now that we know antibodies are biologics, which means vaccines are biologics, other peptides are biologics, so we can use different forms of a protein or a peptide and see how it can be effectively created for treating different diseases. So especially when we talk about biologics, we also think of vaccines and all the other diseases where we can treat novel ways of treating diseases by viruses. Virus can go and kill an organoid or a tumor. So if we create a virus in the space and bring it back to create that opportunity for the virus to kill that cell on the ground, that would be valuable. So that's an innovation right there. And if we sell these viruses and therapeutic biologics derived from the space, I think that would be a property that they attain in the space, not replicated on the Earth. So that's a very novel innovation. So that's where it can contribute big time. As we speak, it has been accomplished. So that's a good story. Next slide. So the key insights into microgravity contributions through Keytruda has been addressed already from the way of telling that at the end of the day, the crystallization matters, and an effective crystal that can interact in multiplanar models can actually contribute. So this drug, when effectively developed, it's a biologic, and it goes and interacts between the tumor cell and the cancer cell by blocking PD-1 receptor. So that means that this protein has to be spatially very effective, very efficient to maneuver through this little space that is available between the tumor cell and the cancer cell. And I think that's a property it gained by having been developed in the space. So that's a very important part to understand. Next slide. So thereby increasing the efficacy of the drug itself. So in microgravity, we can also check not just single drug. There could be spatial interactions which has to be explored. So Keytruda can actually go ahead a little bit further as an innovation, and maybe we can do antibody drug conjugation. So when we add that drug to the small molecule to this bigger antibody, that interaction in the space will happen also in a very spatially defined manner. They're not going to sediment. So all the properties that were exploited by these companies that produce Keytruda and other molecules, as we heard from Antariksh, this can be exploited for creating antibody drug conjugates as well. So that's a novel innovation. I'm sure Sridharan is an expert in this therapeutics of peptides, right? So there is also development of combination therapies, which is important for patients to be cured in a personalized manner. So we can combine different drugs by using this spatial metrics and create a novel drug. So there is opportunity for pharmaceutical companies to test existing drugs. And the most important word that I would like to use here is repurposing. So if a drug is effective for cancer, we can probably combine another drug that is effective for something else and see if that drug could be effective for both the diseases and like that. So we can create such novel molecules, novel therapeutics, and create patents for being created in space, right? So that's important for us to recognize. Next slide. So obviously, when the research is being done in creating these things, there are a lot of factors that go into the cell cycle, right? The cells age because of various reasons. They die on its own. The mitochondria become dysfunctional. There's proteostasis. The telomere gets attritioned or uncoiled in the space, but it is a reversible reaction. There are other genetic manipulations and alterations that can happen. The instability could be created. The cells could be exhausted. So altered intercellular communication and various mechanisms of the cell can probably create a cell that is more amenable to be treated or to be understood as a basic science. So that is a system environment that is created in the microgravity, which is hard to create on the Earth. So even though these sound like cells age faster or alterations in the DNA happen when in space, this could be things that may be desirable because it's hard to create them on the Earth. So that is an innovation where we can look at aging of a cell as an interactive mechanism for how a drug would interact with a cell that is aging in a suspended microgravity where there are substantial forces that keep that equilibrium. Next slide. So I talked about a few innovations, but if you look at Keytruda itself, what happened? SpaceX commercial supply created a monoclonal antibody particle, created a ground-based proof of concept performed, and now we have beautiful suspensions with the desirable particle size that can be used, right? That's a big achievement. Now, if you add these innovations to this achievement, we can create drug conjugates, we can create personalized therapies, and we can understand and address problems of aging, cancer, and many other diseases. Next slide. So microgravity as a tool for protein design has been effectively addressed. It offers various parameters, including convective flows that are also greatly reduced in microgravity, excellent environment to study complex three-dimensional interactions, and it also creates a more concentrated, high-quality mixture. So I'm talking not just about one molecule like Keytruda, but I'm talking here about using these properties in the microgravity to create combination drugs. Next slide. 3D cells are obviously cells that interact together in a suspension and form 3D round structures. And the same properties that are created in the microgravity lets them do it much more effectively because there is free interactive space without the cells going to the bottom of the plate, bottom of the well. So they have multi-dimensional interaction. So the ratio of cells to create an organ or an organoid for transplantation could be effectively done. So it can be brought back to the earth and probably injected back into the site of injury to create a transplant or an organoid for regenerative therapeutic. So that's a novel innovation that can be addressed. Next slide. And hence, there are multiple ways of creating microgravity exposed cells. On the right, you see the picture where it is in gravity, it is settling down, and microgravity suspended. So you get beautiful organoids in the space. And that's our experience as well on the Earth. Creating organoids in a suspension is much more beautiful or easier than creating it in a sedimented plate. So we have to constantly churn them here, but there's no need for it there. And the cell interactions might be much more effective because of the size, the DNA and everything. So that is actually wonderful contributions of space to the cell biology. So people talked about Keytruda, which is a drug. I'm talking about antibody drug conjugates, again, a drug as a combination, and then they're reacting with the cells that are created on the space. Again, a beautiful interactive atmosphere in the space can be exploited. And then finally bringing them back and using as a therapeutic. So we are talking about cell therapy, where the DNA could be modified. We are talking about regenerative therapy, where modified cell can be attached to this damaged tissue on the ground. So next slide. So we can essentially make it very personalized. And of course, the latest, there is 3D printing technology available. Maybe we can put a 3D printer on the satellite. I'm sure RSAT and Jibin and Adeel and all of them are very much aware of it. So we probably can print the organs there because of the spatial interactions much more effectively, bring it back and transplant. So that's another innovation. And then we can create fluid dynamics where the cells don't go attached to the side of the tube or the pipe or the capillary in the space. They may be actually flowing in the middle because the suspension nature. So that's a valuable thing to exploit. On the Earth, this is kind of challenging because you have to force the cells through the tube with a peristaltic pump within the microslide. And that's a little bit difficult to achieve. We are able to achieve it, but it is something that can be exemplified and probably give much more interaction for the cells to form different organs in that ratio and understand the toxicology and everything of those cells. Next slide. Obviously, people have named some of the companies that are working on it. I see future possibilities. If you go from small molecule to the left, all the way to the right, which is 150 kilodalton monoclonal antibody. Now we can create combination of antibodies, combination of these small peptides, which are 2000 or something like that. And we can attach small molecules, which are less than 30, and then make it a super antibody drug conjugate, which cannot probably be synthesized on the Earth, and thereby not even attempting to do that. So a lot of companies doing ADCs are actually challenged by the spatial metrics, because once you bind something, how can they interact with the actual site when that's something that is bound is coming on its way? So those effective interactions can happen in a suspension in the space microgravity situation. Now, if I look at AI that is able to generate the spatial interactions on your computers, now we can create an AI-generated microgravity-based target drug discovery. So maybe AI can simulate how these drugs would interact in the space on a computer model, and then we can create that special thing. That's something innovative that I probably want to propose to people who are attending this seminar. Biomanufacturing is another thing that's in the orbit for ultra pure biologics. So that's the ultimate goal. And I was surprised and happy to hear Antriksh mention that we can actually scale up this biomanufacturing. Manufacturing of peptides, maybe proteins and crystals, all of them could fall into that category where now we can produce APIs in kilograms, maybe biomanufacture biologics in milligrams and grams. So that could be a major achievement, and bring it back to the earth after manufacturing and directly going to the research lab or onto the patient. So that is a therapeutic innovation. Next slide, please. Okay, essentially we can conduct biomedical research in the space. There are technical limitations, however, that many people have pointed out. Cost of transportation, maybe the scaling up of this number of spacecrafts going into may reduce the cost. The experimental flexibility is challenging. Given on the Earth, we have replication challenges of some experiments because people do it in different geographies, different people, different lab setup, different atmospheres. So those challenges continue to remain in microgravity. And the conditions may fluctuate, can affect the biological samples. The cellular responses can be different in microgravity. We have to replicate that multiple times to achieve that consistency. And each batch may be different, so we have to look for quality assurance from those batches. And due to the unique conditions, replicating experiments consistently change. So from spacecraft to another spacecraft, there could be conditions that could change. So these are some of the challenges. So anyway, innovation is not challenge-proof, even on the ground. So I think this is something that we should attempt to do to create novel biologics drugs. Next slide. And further, if you ignore this or solve these logistical constraints, astronauts have limited time. They can stay in the space for some time, so they have to do many experiments in a given time. And it's actually not done by all the experts on the space. So you have to teach one astronaut many things. So that's a beautiful way of really keeping us very active and engaged to create novel molecules. And the communication back to the Earth, how the things come back, how the communication of the results happen. I'm not the expert in that one, but I'm sure AI, computer, and all these solutions that are offered by companies like OurSat and others may be helpful in solving. So these challenges obviously require innovative solutions, careful planning, but also highlight the exciting potential of conducting research in microgravity. And our microgravity situations, environments that are creating novel drugs can then be patented, which is very important for businesses. But of course, creating drugs to cure unsolvable, untreatable diseases on the Earth with novel innovations like this on the space could be valuable. I'm sure Sridhar is very much aware of it, and he is trying to treat rare diseases on the Earth with research on the Earth. So maybe some of these rare diseases could be easily treatable by gene manipulation, CRISPR technology, interactive cells that can be created in the space, brought back and addressed. So there could be funding challenges, but I think innovations are always filled with such kind of challenges. Next slide, please. So this is the lab that I have. It's on the ground, and this is the reason why I'm presenting it is so many instruments, so much space goes into creating organoids. And you need many things to measure proteins. You need FACS proteins. You need ACTA to separate proteins. You need FACS to separate cells. You need cold storage, liquid nitrogen. You need centrifuges to create that cell suspensions and sediments and pellets, storage cabinets, multi-mode plate readers. So if you look at all these things, I mean, a full lab of 2,000 square foot or a 500 square foot would be good for me on the ground. But a 500 square foot lab in a spacecraft or outside in microgravity, that's a challenge. So that itself is an innovation. And I think Adeel and they are the people solving that space constraint. We need to precisely bring the entire lab that I'm showing here on this slide into one small box and then get all these experiments monitored, done, performed remotely, electronically. Huge challenge, but I'm so excited that the people have done it. And it's just such an opportunity for me to interact with so many good expert scientists. But if I can combine my science and Sridhar's science along with the space biologists, I think we are up for extremely exciting novel molecules that can be brought down to Earth as therapeutics for various diseases that are very difficult to treat right now. Next slide. I think that was the last slide. So thank you very much for your attention. I think I had given ample time for Jibin. Over to you, Jibin. Thank you.
Jibin, let me just check where he is. I think he's dropped off for some reason. Suresh, I think we lost Jibin. There he is, back. Jibin, can you hear us?
Hello, can you hear me?
Yes, Jibin. Please go ahead with your presentation. So Jibin is going to talk to us about the payloads, how to take something into space, and what do you do to get them back? So Jibin, over to you.
Definitely. Thanks a lot, Sridhar sir, and thanks also once again, Suresh sir, for your wonderful presentation, and Adeel and Antriksh. I'm definitely among esteemed colleagues today, and most of you, you have covered pretty much most of what I would love to say as well. But I would like to start with emerging development happening in India. As India aims to become a global powerhouse in pharmaceuticals and biotech, a new frontier is emerging, one that takes our R&D beyond the lab and into orbit, where microgravity is unlocking breakthroughs that no Earth-based systems can replicate itself. So this is myself, Jibin, and it's a pleasure for me to present today. Nikhil, can you click on the next slide, please? Nikhil, can you help click on the next slide? Hello.
Jibin, we can hear you, but I think there's been some challenge. Let me see if I can share my screen from my side, bring the presentation.
All good, Dr. Sridhar. Let's we can move on.
Yeah, thanks.
All right. Nikhil, you could keep clicking next, going to my
Next
slides,
slide.
please.
Next slide. Yeah.
Yeah. So next slide as well, Nikhil. So what we have been seeing in the last 15 to 20 years is that the pharmaceutical industry, especially in the US and Europe, have been venturing into utilizing the uniqueness of microgravity environment itself. It might sound like science fiction to you guys, but it has matured. The market has matured there to such a stage that there are four clear areas where the commercial research is focused on. One is on biomedical and life sciences research. One is on pharmaceutical development and drug discovery. The other is on human health and space medicine. And the last one is space-based biomanufacturing and production. These are the four major areas. If I have to generalize the various types of experiments that have been happening over the last 15 to 20 years by various pharmaceutical companies and biotech companies, these are the four major areas. Nikhil, could you go to the next slide? Now, those four areas, if I have to subdivide them down, these are the specific areas of focus: protein crystallization, tissue engineering, aging and neurodegenerative diseases, gene expression and epigenetics, regenerative medicine and stem cells, microbiome and antibiotic resistance, fluid dynamics and drug formulation, biomanufacturing and 3D printing, and a newly emerging topic as well, space-based for pharmaceutical manufacturing itself. It's an interesting fact that over the last 27 years, nearly 3,700 experiments have been conducted aboard the International Space Station, which is floating at 400 kilometers above us, with roughly 45% of those 4,000 experiments focused on life science industry alone. And that just shows that the scientists and the organizations who are aware of the benefits of microgravity have capitalized on it and gone deeper into not just doing R&D experiments, but also have ventured into some kind of biomanufacturing as well. Next slide, please, Nikhil. These are the major companies and entities focused on commercial research: Merck, Novartis, Amgen, AstraZeneca, Eli Lilly, Angiex. Some of the most famous pharmaceutical companies in the world, they have all ventured into microgravity research itself, and they focus on commercial aspects of those microgravity research, whereas fundamental research is conducted by space agencies like NASA, ESA, DLR, JAXA, even universities as well. And so there you see the distinction in the focus. On the one hand, pharmaceutical companies and biotech companies are interested in bringing translational research benefits directly to drug development processes on Earth, or discovering new types of drug formulations in space that can benefit on drug development on Earth. And on the other hand, universities and space agencies are interested in fundamental research or new knowledge creation itself. So this slide is not a summary of all the organizations which are active. It is far larger than what I can fill on the screen, but this gives a snapshot of some of the major players in the microgravity sector. Next slide, please, Nikhil. Dr. Adeel, Dr. Suresh, and Antariksha has all touched upon these points, but if I touch upon some of the biological aspects of microgravity effects, cell shape and morphology, cell adhesion, extracellular matrix, cell division and proliferation, protein production, gene expression, fluid dynamics and transport, cytoskeletal rearrangements, microgravity-induced stresses. These are all known and validated effects conducted over the last 40 years of microgravity research, starting from the Skylabs and the space stations that Russia and US sent back in the 70s and 80s, all the way to even now as well. And these are known effects of microgravity on cells and fluids and materials. Next slide, please, Nikhil. And I want to just touch a bit back to Earth as well. Now, we have talked about how wonderful and advantageous microgravity environment is for scientific research. How do we translate that into experiments that can be conducted? And that's where we come in. So we are an aerospace engineering and manufacturing company specializing in design and development of payloads and satellites for scientific research. So our mission is to enable life-saving therapeutics and advanced scientific knowledge. And that means we want to venture into doing things that have not been thought before, and entering to the microgravity field and offering high cadence and accessible microgravity research opportunities to universities and pharmaceutical industries. As a summary of our capabilities, turnkey solutions, designing and developing scientific payloads, custom designed for experimental needs. Launch support, we have partners with the International Space Station, low Earth orbit launch partners globally, both in India as well as abroad, and suborbital flights in Europe as well. Mission operations, so when these payloads conduct these experiments in space, we can autonomously do experiment management and real-time data streaming from launch all the way to return of the payloads back to Earth. And post-mission deliverables as well, where the samples are retrieved and the comprehensive data is handed over to the clients, whoever they may be. Next slide, please, Nikhil. Some of the technologies that I'm happy to share on the presentation today is our core platform known as ADELAB. ADELAB stands for Advanced Diagnostic and Instrumentation Laboratory. Essentially consists of two particular experimental chambers. One is known as the space microhabitat, the other is known as space fluidic platform. And across these two ranges, we can do a variety of experiments itself. And then we also have a portable centrifuge and mixer vortexer that we designed and developed and tested it as well. But ADELAB forms the core technology that we utilize on every mission, and we custom design the chambers for various kinds of experiments itself. Next slide, please, Nikhil. Like I mentioned, there's a range of experiments: protein crystallization, 3D cell culture research, organoids and organ-on-a-chip, drug discovery, plant growth studies as well. The flight computer systems, the sensor suite, and data acquisition systems, we all developed it in-house, and we have successfully tested it over multiple rocket missions. Next slide, please, Nikhil. Some of the finer details of this is it hosts a microfluidic chip. The microfluidic chip is what enables us to run experiments autonomously, and that's where we host the biological samples. We integrate them with pumps, motors, cameras, sensors, and we package them together as a lab on a chip, and turning the satellite platform into an autonomous laboratory itself. So it has various systems for managing the experimental procedures, and it has two specific chambers: microincubator, microfluidic platform. It is designed to be in the form of a CubeSat, but can be integrated on suborbital rocket flights, ISS launch flights, as well as on low Earth orbit missions as well. Next slide, please, Nikhil. If I touch on the microincubator, it's focused more on stem cells, 3D cell culture research, and protein crystallization. So there's cameras with 400 to 800x magnification, sensors, standardized sensors, and also experiment-specific sensors packaged along with it, and as well as microfluidic chips hosting the living cells, so that we can allow them to grow, we can allow them to be incubated, and also preserve them and bring them back. Next slide, please, Nikhil. The next chamber is a fluidic platform, which is more catered towards the drug and pharmaceutical research, fluidic control, complex fluidic dynamics. That's the core capability of that particular platform itself. And that, in addition to the standardized sensors that we usually put on experimental chambers, it has a fluidic network of macro reservoirs, macro pumps, and a fluidic network to enable us to run experiments in a autonomous manner. Next slide, please, Nikhil. How do we work? How do you work with the different clients, and how is microgravity research even possible? So someone has to take care of the regulatory approval. Someone has to take care of the engineering, designing, and development of the payloads. Someone has to do the rocket launch booking and put the experiments on the rocket to be sent to space. Someone has to stream the data and bring the data and the samples back down. That's what we do. We take care of the end-to-end services, which involves not only just the product that we offer for the research, but also the various other aspects of these projects, which might be outside of the expertise of the clients who come from a purely scientific background or from a commercial non-engineering background itself. And that's where we take care of all the regulatory, engineering, rocket launch management, space mission management, and experiment data and retrieval itself. Next slide, please, Nikhil. We did our first suborbital launch in November of 2022, where we hosted our payload on a suborbital rocket from Sweden, where the payload was sent to 257 kilometers. It was a suborbital flight, and suborbital flights have short duration microgravity flights. It's usually used by scientists and by universities to do a very preliminary, simple experiment to validate the effects of microgravity, whatever is possible to observe, and then developing a hypothesis on that to go for long duration flights on ISS missions and low Earth orbit missions itself. So this suborbital flight we did where we hosted one drug delivery client and one university, and we ran experiments covering both drug microencapsulation as well as on microbial nanobiotechnology and microbial research. Next slide, Nikhil. So the fluidic platform hosted the drug delivery experiment, the drug microencapsulation experiment. And on the picture itself, you can see that the microgravity chip generated emulsions, water emulsions, so that the microencapsulation experiments could be you could package the drug molecules with membranes and run experiments on them. And on the microhabitat platform, you can see on the left Earth samples on the yeast cells that we grew on Earth and the yeast cells that grew in space, and which seemed to indicate the formation of what appears to be the formation of a biofilm. And they grew 4 times faster and shrunk down in size. Next slide, please, Nikhil. The second suborbital flight, we did it last year in again from Sweden. This time, the German Space Agency, DLR, was the main rocket supplier, and we hosted our payload in the picture, as you can see on the top right. It was hosted there with cables coming out for data transmission wirelessly. So antennas on the either side of the rocket module itself, which is visible as those hooks. And again, we ran it for a suborbital flight for different clients who were interested in mostly on gut microbiome research, and another company who wanted to do technology demonstration flight itself. Next slide, please, Nikhil. And for the technology demonstration company, they are a non-biological company. They just wanted to use our platform as a hosting platform to test their systems. And the actual client, Cambridge Executive, for them, we did microbial research experiments across 3 different samples hosted in cuvettes. This was a passive experiment, so no cameras were looking down on the samples. They were passively hosted and allowed them to grow and brought them back, experiencing the range of conditions from sea level all the way to microgravity and then brought back down. Not to mention the violent rocket launch conditions during launch and return itself. Next slide, please, Nikhil. So our platform can go on various types of missions, and that's very important to know because different kinds of scientific research requires different kinds of parameters and hypotheses that they want to validate. So we talked about, we heard a bit earlier about Antariksh talking about his reusable satellites, which would be sent and which would function more like a low Earth orbit mission. Then there's ISS missions, which is what we are looking to do next. And there's a suborbital missions, which we have done 2 times in the last 2.5 years. The difference between, broadly speaking, the main difference between these 3 opportunities is the microgravity time. So you get 7 to 10 minutes in suborbital flights, you get 3 to 6 months in ISS missions, and you get 6 to 12 weeks on low Earth orbit missions itself. And that greatly influences what kind of complex experiments you want to run, what kind of parameters do you want to design the payloads for, and what are you actually looking to investigate for. Next slide, please, Nikhil. So this is where I just want to run a little bit more detail into the payload development cycle itself. For example, we are looking to do an ISS mission in February of next year, where we are inviting interested universities and industries to participate on this launch opportunity, where we'll package their experiments and send them to the ISS itself. Next slide, please, Nikhil. So let's say we want to do protein crystallization experiment in space. How does it translate from the research all the way to space and bring it back down? So it becomes a conceptual understanding of what the objectives, goals, and outcomes would be. let's say we want to do a microbial research experiment itself. And then within microbial research itself, what microbes do you want to select? What are the experimental specific systems that you want to host for monitoring them? What are the growth conditions and parameters that need to be investigated? And cryopreservation of the samples to bring them back down. Next slide, please, Nikhil. Once this is done, then the entire payload is repackaged into incorporating various kinds of engineering systems that can run the experiments. And then the entire payload can be sent to US for integration and launch and so forth. Next slide, please, Nikhil. Before it goes to the US or before it goes to the launch site here in India, on a parallel phase, we do experiment conceptual ground studies on the one hand, we do payload engineering development on the other hand, and we work hand in hand with our clients, both with universities and industries, to ensure that the entire research translates from conceptual design all the way to flight-ready hardware that can be sent on rockets. Next slide, please, Nikhil. Then it is sent to US for qualification and acceptance testing, which requires a certification that your payload can be sent to the International Space Station as an example. And 48 hours before launch, they would put the payload and the biological samples in deep freezers and then prep them for launch. And then they will be, SpaceX would send its Dragon capsule to the ISS. Next slide, please, Nikhil. And that's where the ISS module will dock with the International Space Station. And next slide, please, Nikhil. And the astronauts will take the payloads out and integrate them and install them in experimental racks. These are called ISS Express racks. This is illustrated a picture of how the payloads that we send, how they'll be packaged and integrated. As long as the payloads are integrated with the ISS Express rack, we can do data streaming of the samples itself. Next slide, please, Nikhil. And the samples data can be streamed down, the images and the videos can be captured and displayed on a web-based portal that interested parties can go and investigate it and see how the experiments are monitoring and how they are growing as the ISS mission goes along. So it's a 1 month long flight, and hourly basis, the data is shared down to Earth. Next slide, please, Nikhil. And as a summary of an ISS mission, typically, if it's a very first engagement that we're doing with a particular type of experiment that has not been designed before, it takes about 6 to 8 months in the ground phase. And then it takes about 1 month long ISS flight. And then it takes about 1 month long for the payload, not only to, it takes just a few days for the payload to return back to Earth, then to take it and then bring it back to the host laboratories or the destination laboratories. That takes anywhere from 2 weeks to 1 month. Next slide, please, Nikhil. So this is where ISS opportunities are there, and this is where we are currently working on as well, where we are offering active experimentation, passive experimentation, cell bank experiment, in-space manufacturing. There's a wide range of experiments which are of definite of interest within the Indian ecosystem, and I would love to see our countrymen, our experiments going on the ISS mission in February of next year. And that's one of the things which I just wanted to highlight here that we are working towards, and we are calling out to different universities and industries as well. Next slide, please, Nikhil. And this unlocks the entire opportunities for science and business, whether it's on scientific side of things, where you get precision-driven experiments and insights unattainable on Earth. Technical side, where it's advanced instrumentation, AI-driven analytics. Pharmaceutical side, where it's better drug efficacy, new formulations and faster R&D cycles. Commercial side, you get competitive advantage, IP generation in a frontier market, and as well as branding and storytelling as pioneers in space research. Because like I mentioned earlier, Southeast Asia is nowhere near as whereas the US market is or the European market is. They are pretty much running away with what they have found. And it's one of my desires to bring India and our ecosystem to the global competitive level itself. Next slide, please, Nikhil. So what is emerging beyond the R&D experiments? The in-space manufacturing, and some of us on the call to touch upon it as well. There's the microgravity R&D sector, which began in 2005. Now it has matured to in-space manufacturing of new types of fiber optics, solar materials, batteries, e-mobility materials, and the biological side of things, new larger kinds of protein crystallization, as well, which Antariksha was talking about, as well as stem cells and stem cells and tissue engineering, which can be generated in high-quality environment and brought them down. That's the emerging in-space manufacturing sector. Next slide, please, Nikhil. This is a snapshot of some of our team members. Myself and Ravi we both were classmates at University of Adelaide, where we did a master's engineering in aerospace engineering, and that's where we developed the concept. We have Saki and Srishti who form part of the executive team, and we have a few advisors who come from both from financial perspective, from the medtech pharmaceutical side, and along with Dr. Suresh, who it's a pleasure for me to have him on the call today, and Dr. Adeel as well, who's a space biologist, and wonderful to have him on the call today to talk about microgravity research opportunities. The last part I just want to touch upon, next slide please, Nikhil, is our Indian company that we incorporated in Hyderabad, active since mid of September, where I function instead of the CTO, where what I used to be doing in research sat, but within our sat, I'm functioning more as a CEO, and there's Rahul and Shrishti and Ernest who's forming the core team, and we are pretty much offering the same technologies and the same opportunities, custom satellites, turnkey logistics, and research, and new kinds of research for biotechnologies, pharmaceuticals, universities, and ISRO too. So the microgravity research is no longer science fiction. It is available, it is here, and it is now. And we have wonderful people on the call today, and we have companies emerging in India who would love to work on this further. Next slide, please, Nikhil. And that brings to the end of my talk. It's a pleasure for me to share this, and I hope you have enjoyed not just my talk, but also all the other panelists as well. Thank you for listening.
Thanks, Jibin. Thanks to all my panelists, Adeel.
I think you
Antrix,
muted
Suresh.
Dr. Sridhar.
No one.
Yeah.
I think I should be audible. Yep. Thanks very much, Jibin, and thanks to Adeel, Antrix, and Suresh for wonderful talks as well. We have about 10 minutes for questions, and we already have quite a few questions in the chat box. So I'm just going to pose them one by one. So one of the first questions was, how does the integrity of the protein crystals and organoids hold up once they are brought back from space to Earth? Antrix, you want to take that one?
Sure. Thank you, Dr. Sridhar. So, yes, they actually notably have had a good history of survivability when they have especially returned aboard, let's say, the Dragon capsule, or even if you're following a company called Varda Space Industries as well, they also landed their capsule back, which was an unmanned capsule, and the protein crystals and, in general, pharmaceutical crystals show good, basically strength during it. However, they need to be, when you're coming back in a manned capsule versus an unmanned capsule, the challenges are different because, of course, there are different levels of G-forces during re-entry, depending on the geometry. But then how you actually bring it back, and can you maintain the conditions of temperature? Can you have something like a suspension which can basically neutralize shocks and vibrations? Those are going to be essential. Another like innovation could be that you can freeze them, but often what happens is protein crystals lose their mosaicity and structure when you freeze something like that. So freezing is usually not done unless it's a very robust or a simple crystal. But mostly it is like you can sort of, if you guys have seen Colgate, you can sort of embed them in gels so that you restrict their mobility and then you can bring them back. So that is something that is usually done, and then it survives.
Thanks, Antrix. Adeel, one question for you. Will a drug formulated in microgravity have a longer shelf life than a one formulated on Earth?
No, basically the simple answer to that. The conditions of space, actually, I think I should have put that part as well, are reducing the shelf life. So the challenge is for working in the microgravity research that how we can improve the shelf life. Once it goes up there, the knowledge that we have is from the low Earth orbit. Going further to this radioactive zone, I think the drug stability will be an issue in coming days.
Can I add something to that, Sridhar or
Please
Aditya?
go ahead. Please go ahead.
Yeah, so I actually, yeah, that's true for most drugs actually. But then when you're making a formulation, I actually studied the interferon alpha paper that was done in late 1990s, and they could actually increase the shelf life because they could, of course, change the formulation properties, but they could increase the shelf life sixfold. Compared to the same suspension or the same crystalline suspension of interferon alpha that was done on Earth. So there can be, in case of biologics, there can be improvements where shelf life can be enhanced. But in general, if you leave it up there, of course, there's radiation. So largely, Dr. Adil's answer is correct, which is why you need routine re-entry. You cannot wait for like 6 months and just leave it, and then the next batch of astronauts gets it. So you need routine and quickly coming back. But yeah, just wanted to point that out, that increase in shelf life is also possible, and it has been done in the late 1990s. But that was just one.
So thanks, Antrix, for adding that on. My fellow panelists, request there are questions in the chat box which you can directly answer as well. If you can type in your responses there, but I'll ask a few more questions. In the interest of time, just going to limit those. One of the questions, and Suresh, you might be able to answer this one, is there advantage to doing sterile manufacturing in space?
Yeah, excellent question. So the sterility problem on the Earth is due to numerous contaminations in the cell culture hoods and everything. So while when you miniaturize the whole manufacturing, the chances of sterility, keeping it sterile, is very high. So in the space, you don't have other organisms able to survive due to radiation or other things, so there's contamination chances are less. But what goes in the tube goes there. So if you send it with an contaminated mycoplasma or something, that also will proliferate faster and really spoil the culture up there. So sterility is virtually easy to have in the microgravity up in the space because of the limited quantity and the space constraint, but difficult or very impossible if you actually send in a contaminant along with it. Yeah.
Okay. The next question, and I think this is a really out of this world question, is can we treat a terminally ill patient better in space rather than on Earth? Open it to the panel, whoever wants to take that one.
I'll leave it to the scientists to say.
I would make some speculations, Sridhar. I think hopefully if you agree with me, I will be happy. The thing is, there are numerous cellular changes that happen in the space. So that has been studied in, I think actually I was having an interactive discussion with Adeel yesterday. I learned this as well, a couple of things from Adeel. So the cells grow a little bit faster in the space. So there is a healing ability that is possible, number one. Number two is they try to act like they are proliferating fast and mature, but when you come back to the ground, they may keep that property for some time, and then they may develop additional properties that may actually enhance the healing by way of producing extracellular matrix proteins like fibrins and other things, right? So there are matrix proteins as well as extracellular like glycans and those things. So those can be extensively produced, which may lead to a healing. So based on what kind of disease we are talking about, it could probably enhance the healing. If you are talking about cancer and if you send somebody there, the cells proliferate faster in space. So that may not actually heal it. It may kill him faster. So regenerative diseases, absolutely a good way to send somebody to heal, close the wound, close the sutures, maybe heal the tissue, regenerate. But for cancer, I think I would stay away from that.
Yeah, just a footnote to just touching on the cancer part itself. I came across this video from BBC itself where I forgot which cancer cells they were talking about, but they were talking about how they had this particular cancer cell, which cancer cells, which doubles in size in about 10 years in time. And in space, it happened in a matter of a few days. So it doubled and grew in size very, very quickly. So yeah, just touching on that, yeah, it's very fascinating to know.
So thank you, Jibin. Thanks for substantiating that.
I think it reminds me that famous movie Contact, where a billionaire was giving the money to the scientist to go for build that machine, and he was himself suffering from cancer. And he was trying to see that if space can heal him. Unfortunately, his dream wasn't became reality, and neither anyone who's planning to go up there to get healed, this won't happen, to put that debate to end. But there are some interesting things happen when we are in that environment. For example, that Dr. Suresh just talked about the book from Dr. Christopher Mason, where he's saying that two twin studies, Scott Kelly and Mark Kelly, became a parliament. I think he became the member of the parliament afterwards. So the fact is that he had some changes. His telomere increased. And for a gravitational biologist, this could be because of when you do not have the mechanical force pulling you down, and you're actually basically, it's just like protein structure. All those structures that we have, they go in that free environment where you don't have a force pulling you down. For the time being, you may have those temporary changes. And we haven't started studying the evolution of human body. And it's a very short-term exposure that have perhaps, if we expose, I mean, this is a crazy experiment, but we might see changes and adaptability to that environment, but that adaptability environment would stay for the low Earth orbit. If we go to Mars, the gravity would change. Again, life has to readapt. If we go to Moon, the gravity would change. We have to readapt. So I think there is not fit for all for everything. We see some changes. For example, cancer cells. There is a new group of scientists who are focusing that cancer cells in microgravity, they are not as aggressive as they are on Earth. The absence of gravity does something to that, and they're trying to repurpose drugs, that if in that less aggressive form, we can check if the drug can be used, and we find the target in the cell, in a cancer cell. So I think there is a lot going on, but to say that, we did not evolve for adapting in space, and we should not expect that space is such a beautiful place to live. It's a harsh environment. So we have to keep that in mind. We should not make it like a hunky-dory. It's not a fun place to be. It's not, there is no place like home, Earth. So that's why it's the responsibility. As a scientist, we should protect it. Wars, hunger, and environmental problems, these are our enemies, and we should, celebrate that. So we should stay away from that. And I think for going, now it's like a commercial, it's open for commercial audience. Again, it's, billionaires playing in that. So I don't know.
Thanks, Adeel.
Yeah.
I again, a request to my panel members. There are at least another 15 questions on the chat box. If you guys can take shots at answering some of those, I would really appreciate that. For the participants, thank you very much for being a part of this and asking so many questions. If you want to contact any of the panelists, do feel free to write to me. You would have received emails from me regarding the webinar itself. Happy to connect you to each of them. Today, this has been our first attempt to bring a very new technology to an audience in India. I believe this is the first webinar of pharmaceutical research in space that has ever been conducted in India. And I'm really glad that through IPA, we've managed to do this. In addition to companies that are part of this panel, there are several other companies which continue to work on this. A few of them, like Inbound Aerospace, Metacosmos, bring very interesting technologies as well. So if you're interested in space biology, in space chemistry, and pharmaceutical research in space, I think this is the time. India is really putting an effort to build its space presence. And I don't think there is any better time than now for us to exploit that to the best of our abilities. So thank you all very much for the excellent presentations, my fellow panel members, and to all the attendees for being part of this session. Thank you very much. And before I go, thanks to Nikhil, my colleague from IPA, who's been instrumental in making sure that the presentations were proper and all of you received the web links on time to join. Thank you very much, Nikhil, and thanks to everyone.
Thank you, Dr. Sridhar.
Bye.
Thank you all the panelists.
Thank you.
Thank you so much, everyone.
Bye, everyone.
This was
Thank you,
fantastic.
Sridhar. Wonderful
Very
job,
nice to meet you.
Nikhil. Thank you.
Thank you.